I will present this session along with my colleague, Ethan Sanchez. So there are some general announcements to make. Please use the Q&A panel to ask questions. And if you want to make a comment or ask or speak for yourself, so raise your hand so one of the panelists can allow you to unmute you. And after the demo today, attendees can share that any results or experience from the explorations they tried before. And also don't forget to leave a feedback using the link in the details page. So in the next 45 minutes or so, I will briefly walk you through an introduction to the graph database system, especially the Neo4j database and Cypher, the graph query language. The objective here is to provide you with enough background so that you can understand the significance of our choices and also the Cypher syntax so that you can follow some of the use case demonstrations that follow in this presentation. Next, I will talk about the CTM2, the cornerstone for data discovery in the CFDE ecosystem. And also the interactive graph query system that Ethan has been developing. And finally, the last 15 minutes or so, Ethan would give you a use case demonstration. So before we venture into the graph database system, let's review and understand the limitations of the current relational database system. Though in relational database systems, the data is organized in tables. And even though the term relationship is in the vernacular of the RDB, but it is not modeled explicitly. And in fact, they are inferred at the runtime using join tables via the primary and foreign key associations. This has an impact in the performance in large data sets, and also it gets worse in connected data sets like the CTM2. The second limitation is the flexibility. RDBs require a stringent, rigid schema requirement, which is very unrealistic when you're starting a project. And especially given the dynamic nature of CFDE, where many DCCs or the common fund programs enter into the ecosystem with every year. And also the current common fund programs require new metadata to be introduced so that they can better annotate their data. So this imposes a great challenge. The schema is not flexible, it's very rigid. And the most important one is the performance. The queries are really expensive. This is primarily due to the underlying data model, which imposes the data normalization process. Where what it does is it keeps breaking the data elements into its own tables in order to minimize the redundancy by introducing dependency. So at runtime, we need to reverse this process of normalization to denormalization by joining tables together, which results in large data sets and become very unwieldy with large and interconnected data sets. And the queries can become really expensive. For example, it's easy to ask a question, what products a customer purchased? Whereas if you ask the reverse reflexive question, what products, given a product, what were all the customers who bought this product? It becomes expensive. And also the recursive queries like what customers bought and what they bought, something like a sort of a friend of a friend kind of questions become really expensive in relational database systems. The other limitation is the agility or scalability. Relational databases are vertically scalable, but they're not horizontally scalable. I will talk about this in a little later. So this is the current CTM2 entity relationship diagram. It has got about 49 tables with lots of, highly connected and metadata. And right now it is about more than 1 million records, and they are expected to continue to grow with the time. And so there is obviously a need for a database system that is schemaless, flexible, agile, and most importantly, the performance should be good or responsive. So this brings database system as a natural choice. And a graph is a collection, mathematically, it's a collection of nodes and the relationships as an ordered pair. Here you can see the nodes N1, N2, N3, and relationships are ordered pairs. N1 is the start node, N2 is the end node. And in graph, the entities are represented as nodes, and the relationship between the entities are represented as relations. A graph data system exposes and underlying data to CRUD operations, create, read, update, and delete operations. And there are three attributes that characterizes a graph database system. One is the model, and data storage, and processing. And each graph database system is unique in that way in how they impose or handle these three entities or three attributes. Some of the graph databases use LPG or labeled property graph. Some of them use RDF, and some use hypergraphs. And this affects the way how the modeling of the real world complex scenarios. When it comes to data storage, some graphs, database systems store them as a graph structures like node, relation, and nodes. Some of them serialize into subject, object, predicate, and some of them as general purpose stores. And this affects the flexibility or agility of the database system. And it comes to the data processing. Some systems use native data graph processing. What it means is the connected nodes, they physically point to each other. And if you want to look up, all we need to do is just do a one lookup of the starting node and follow the relationship to find the end node and pull the data. And this has a tremendous impact on the performance. And here are some of the examples of the graph database systems: Neo4j, ArangoDB, and Neptune. And our choice for the graph data system for CFDE is the Neo4j because of its unique advantages. Number one, when it comes to the data modeling, it uses labeled property graphs. And they contain nodes and relationships. And nodes can contain properties and labels. For example, the subject node has a label of subject, and the property is ID, age, and sex. And the relationships are named and directed. In Neo4j, relationships are first-class citizens. They have the type or the name or the label, and they have a start node and the end node. And they can also have properties. For example, here, taxon ID is a property of as subject. And the labeled property graphs are very intuitive and powerful, so we can model most of the complex real-world situations. And they also allow global graph algorithms to run on the graph structures to draw insights. And they are very additive, meaning we can remove or add any node or relationship or the properties, and they are quite flexible with changing needs of C2 and M2. So this is the current ERD diagram of the relational database, and this can be converted into a very visual and intuitive graph database model, where you can see the entities and the relationship explicitly modeled. For example, here this schema has some of the administrative nodes like DCC and ID namespace, some organization nodes like project and collection. And the core entities in this model is subject, file, and biosample. And subjects can have the annotations of a disease or a phenotype. They have a species annotation. And also they can be treated, the subjects may be treated with a drug compound in the experiments. And similarly for the biosample, they are derived from a particular subject. They have anatomy attribute. They can also be inherit some of the attributes from the disease and the phenotypes attached to the subject. And the biosample can be perturbed with either gene perturbation or chemical perturbation in the studies. And the file has data type, the format, and it is generated by a particular asset type and generated by an analysis type. These are the attributes of the file. And we can see the entities and the relationships very explicitly. It's very intuitive. And what seems to be a whiteboard model becomes into a real physical model in Neo4j, the labeled property graph. So when it comes to the data storage, Neo4j uses native graph storage, where the nodes relationships are stored as a continuous structures. For example, here we can see a small graph. A person owns two properties, and he lives at one, and the other one he rents to another person. So we can see the relationships have start node and end node. And the nodes may have incoming or outgoing relationship, not necessarily both. So this helps us to model any real-world complex situations quite faithfully. And also it optimizes the storage and managing the graphs in Neo4j. It's easy to add or remove a node, a relationship, or the properties at ease. And so it is more flexible and agile with the changing dynamic nature of the CTL too. Unlike the relational databases or some of the no schema databases, the relationship is not explicit. They are input at the runtime. And non-native graph models like the RDF resource description framework or hypergraphs, they serialize the data into relational or some object-oriented model or even as a general purpose data stores, which is not good for data storage. And of course, there are some workarounds to mitigate these issues, but not without the risk of data loss and some expensive solutions. So when it comes to the data processing, Neo4j uses native graph data processing. What it means is they connect the nodes which are connected to the relationship. They point to each other physically. When you want to look up for a particular node, all we need to do is a one lookup with the ID. Again, this process is minimized by the label type or the relationship type. So it basically segments or localizes your search to a small portion of the graph. Once you identify the ID of the node or the relationship, all we need to do is just follow the pointers to find the next node and pull the graph out of it. So this is called as index-free adjacency, which gives a tremendous boost to the performance. It enables a fast transversal of nodes, for example, about 2 to 4 million nodes per second with a single core. And the order of performance is the first order, irrespective of the data size, because the search is localized to a small portion of the graph. It doesn't matter we have million nodes or billion nodes. To pull a graph structure, it's always going to use the same number of steps. And the cost of traversing n steps or degrees deep in the network, for example, friend of a friend or friend of a friend of a friend, 2, 3 degrees, the order of operation is going to be order of n, which is proportional to the database size. When you compare this or contrast this with other database models like the SQL or NoSQL, which all rely on lookups or document linking, or some of them even extract a subset of data and pipe it through an external processing framework like MapReduce, which introduces latency and the performance is really poor. For example, for simple queries, it is in the order of log n, and for traversing m degrees deep into the network, it gets even more expensive n log n order. If there is time, I will show some benchmarks of comparing between the Neo4j and other graph database or relational database systems. So this is how the data is stored in the Neo4 data structure in the memory. It has got two large arrays. One is a node array and there is a relationship array. The node array has an ID and it has got the types. And the types are grouped by, they are all typed by the group. And each node has relationships which are typed by the group. And they have incoming relationship and outgoing relationship. And also there is a key array, which are basically the key value pairs. And in the relationship, again, the same ID, and it has got the start node and the end node and what is the type of relationship. And then they have an array of key and value pairs arranged in this way. So what it means is to look up, for example, if you want to look at the person, Tom, you want to look at this particular ownership relationship, all we have to do is go find the index lookup for Tom. Once we know, and the relationship type is there, it has got two types. One is owns and leaves that. We are interested in owning. So let's say the owns is type 1. We need to go to type 1 and look at the R1, what is the relationship? And it's basically a pointer to the relationship array. And then it tells if you're coming from what is the next node. It gives you the next node ID and go back and then traverse. Okay, if this lives at the another relationship R2, then you follow. So the pointers become we use pointers instead of lookups. It has got a fixed size record, joins on creation, not during runtime. And this goes in a loop until you achieve your task. So this becomes really, really fast. And so the coding Neo4j database, I've just briefly introduced the Cypher and the syntax. Cypher is a declarative query, graph query language that enables CRUD operations on the underlying data. And it allows expressive and efficient queries on the graph database. The basic query structure looks like an ASCII art, a parenthesis for a rectangular thing, an arrow pointing to another node. The nodes are represented in round parentheses, relationships in the square brackets with a directed arrow. Either the arrow can be from left to right or from right to left. So this is the start node, relationship, and the end node. The query is basically a pattern matching. We are asking the Cypher language to match a particular pattern and pull the graph. And most of the syntax is inspired by SQL, Haskell, and Python. So it becomes very intuitive and easy for people who are working in relational database to migrate to Cypher. So the nodes are represented in parentheses. For example, here I am representing the node subject. And the node has properties. We can see here it has got an ID, age, and sex. So the properties are key value pairs, which are in the curly braces. And we can assign a variable to this node by putting a variable and a colon. Now the subject node is assigned to a variable s. And if you want to access some of the values, all we need to do is the variable dot the property in the key, and that will pull the value associated with the key. The relationship is represented in square brackets. For example, has subject, study has subject. And this is the way it is represented. Study, which is assigned a variable st and subject s and as. And we can also assign variables to the relationship. And we can access the relationship property by r.taxon ID. That would pull out the value for the taxon ID here. And this example query, it returns the studies belonging to the project. Say project produced a study, and where the project ID is, this is the filtering clause. If you provide that, it will return the particular project associated with this relationship. So a Cypher query is nothing but just a pattern matching. You can use, this is the fundamental one. We can cascade or concatenate the relationship to extract or build much complex queries with further classes. So this is the deployment. The Neo4j, the database is running in a Docker container. The front end is a React.js application, and the queries are forwarded and the response is written back to the application. So some of the use cases, we classify the use cases into search category, discovery. Search is when the user knows exactly what he's looking for. Let's say if he knows the project ID or a study ID or a particular pattern that he is looking for, he can come input those parameters and can get what he wants. That is the search criteria, search use case. The most important or exciting one is the discovery part, where it's called a serendipitous discovery, where you come up with, you have no idea about what are the assets in the CFDE, and if you want to come and discover what are the data sets available that that interest you. So this is where the Neo4j database schema, which is very visual and intuitive. By looking at the schema, one can traverse the graph visually and frame the queries and pull the results they want. For this presentation, we have some use cases. For example, to find all sub-projects associated with the project. And second use case is to find all human lung cancer studies. And the third one is diaptic studies conducted on dog blood samples. The fourth one is a gene expression profile in leukemia patients from LINCS. We are planning to implement graph analytics, where you could invoke graph algorithms on the graph data set to find, for example, centrality or community detection and things like that, just not implemented yet. And finally, we also planning to implement something like a product recommendation. If you're finding a study, what are the similar studies that are present in the data in the CFD ecosystem, and kind of a product, suggestions and things like that. I just want to say we are still in the very early stages of this development process. We just barely got things to work. So it's still in the evolution process. So there will be a lot of bugs. So we encourage users to be patient and try what they want to do and report back the feedback. It will be appreciated very much. So the first use case is find projects. So for example, in this Cypher query, it's very intuitive. So match, we are asking the Cypher query to find a pattern. Project is parent of a sub-project. So basically a project contains other sub-projects. And we are using the filter clause where the project name is Metabolomics. And give me all the distinct project and the sub-projects. And this is a screenshot of this use case. So we have the Metabolomics is the root node or the parent node. And it's got all the sub-projects associated with this parent project. The second one is the human lung cancer studies. Here in the first 4 lines, what we are asking is, we are asking to find subjects whose species. We are matching a pattern, a subject associated with a taxonomy. And then the subject should be tested for a particular disease. And the filter class is the species should be a human and the disease should be cancer. So the first 4 lines or 5 lines, it asks, find human subjects with the disease annotation of cancer. And then once you find that, once you collect that, then we do the second one. We want to identify the projects that contain those subjects, which has an annotation of lung cancer, and give me those. This is the screenshot. So we can, if you look at this one, this is in relation to the previous query. So we are asking the subject associated with the taxonomy. I'm sorry. Subject associated with the taxonomy, subject associated tested for a disease, and then you find those subjects. Yes, Neela.
My question was, when you look for these keywords like lung cancer and cancer, are you looking at the whole study, like the entire description that's there, like the study summary, project summaries, treatment, whatever, yeah, all those different fields, or are you looking at some specific part of the study?
Right. So it's all coming from the C2M2 metadata schema, whatever the annotation fields and whatever it is filled. So it is not like in Metabolomics Workbench, you have the project summary and things like that. So whatever the metadata fields, it's filled with. Every project has got the title, ID, and a short description. So if the description has that, then it can provide. Or if the description is annotated with a disease condition and those things, so that is where the C2M2 schema comes into play.
Srini, there is one more question, so I'm going to repeat the question, or if you are able to see the chat, you can see. Question is, graph databases have many great features, but they also come with known drawbacks such as complexity in querying, scalability issues, data modeling challenges, etc. How are you dealing with these challenges, which have been the main challenge so far?
Okay. Yeah, I can talk about that. But yes, it is not a panacea for all the ills of a relational database or other databases. It has got its own baggage. For example, if you are trying to do some kind of an aggregate, if you're trying to compute what is the average or how many hops are there, something like that, then it lags, because when you look at the data structure, the key value pairs can come in any order. Whereas the lookup operation is very efficient when you try to go and bring the properties. If you're trying to look into the properties, they're not aligned in a particular, because it's schemaless. It can be, let's say, for example, how many subjects are there in a particular study? That property can be in field 1 for one study and maybe the same information is there for another node in 30th key value pair. So it lacks, if you're looking for a relationship within the data, if you want to pull that, yes, it's an excellent tool. But when you're trying to compute those kind of things, yes, it has got drawbacks. And regarding scalability, they are horizontally scalable. And the Neo4j has come up with, maybe I can show that slide at the end. It has got scalability, solutions for much better than compared to the other database systems. So one more hand, Sashi.
Yeah. My question is very basic in the sense that this query that you have right now on the slide there. I mean, how, in order to generate that query, is it done through some kind of an interface, or is there some template or something like that, or how does that work?
Okay, so that's exactly what we're trying to do. I mean, even though the Cypher query seems to be intuitive and easy to learn, but you can't expect a general user to know the Cypher query language to come and interact with that thing. So we want to move away from the Cypher querying. We want to create an interface where people, if they look at the schema, and you should be able to interact with it in a natural way. That is what Ethan has been working on. So the reason why we put this is to understand people. So writing, even though it seems simpler, but you can't expect people to learn Cypher query language to come and interact with your database. So that's why we are developing to get away with the Cypher query visually. How do you interact and pull this? So that is what Ethan is going to demonstrate.
Okay. Thank you.
Yeah. So yeah, I mean, the things can go on, but it's better to, because of the sake of time, as well as, let me show some of the scalability. Okay. So the question about the scalability, the Neo4j is horizontally scalable by sharding techniques. You can break up a giant database into smaller things, and you can run them on autonomous clusters. And once you do that, it also enables a federated query. The users can come to the portal, send a query, which can be forwarded or federated across different clusters, and you can get the results. So the scalar, it is horizontally scalable when compared to relational database systems. But the other non-SQL database systems like White Column, they handle well with the scalability issue, but they are not expressive enough. If you are dealing with a highly connected data, your best bet would be a Neo4j. Since we are running out of time, I will hand over to Ethan to show the demonstration. And if you have any questions, please ask them at the end. We can answer those questions as well. I'll stop here.
All right. Good morning, good afternoon, everyone. My name is Ethan Sanchez. I'm a software developer with the Superminium Lab. I've been working on this interactive graph query interface. Srini already showed some screenshots from the interface and also shared some Cypher queries explaining how to produce the results in Neo4j, but I'm going to show how you can produce those results through this interface. First, the way this will be structured is I'll sort of show the results, and then I will show how we generated the results. So this set of results should look familiar. Srini just sort of described, we have this Metabolomics Workbench project, this root project, and then many other sub-projects that are contained within. We can see they're all connected by this is parent of relationship. So how did we produce these results? We have a tool here where we can essentially build like a Cypher query, but in a hopefully more intuitive way. So just starting off with what we have here, we can see that there are sort of three elements here. We have a node, which is this root metabolomics project. it has a is parent of relationship to some subprojects, which we've called MW subproject. We set a limit to 50 here. There might be more. And if we were to, let's close this, we do our advanced search, we'll see that it searches and we come back with these results here. So just to come back, I know this was already sort of created here. So let's just do this from scratch quickly. So I'll remove this path. We can see we have this add a path button. Click that and we have a sort of blank search field. If I click on that, we'll see a drop-down that has a list of the many different types of nodes. And also, if I scroll down, the relationships that are in our graph database model. But again, we're interested in projects of projects, so I'll click project. And then we can see that this drop-down is sort of contextual. It will adapt to whatever relationships and nodes are actually connected to this project. So in this case, a project can contain biosamples, files, subjects. It can also have children. So project, we can see again, is parent of. We can see also that it's connected to DCC and namespace through some other relationships. But we're interested in this is parent of, so we'll go ahead and click that again. And then again, interested in subprojects. But we're specifically interested in MetabolomicsWorkbench. So we'll click on this root project. We can see we can add a key here. This is essentially just a variable name. It's not particularly important in this example. What we're really interested in is adding a filter. And we're going to add a filter on name. So name, it's a little cut off here, but we see name equals, and we're going to say metabolomics. And if we run this again, I'll just set the limit a little bit higher again so we can see that nice group of projects. If we go ahead and search, and we can see the same sort of result here. So this is what we're hoping to do. This is a very simple example, just going from one node to another. But you can see that query that Srini had earlier was sort of 3 lines of Cypher, and we've turned it into just one sort of simple interactive UI element here. It's just 3 elements. So that's a very simple example. We can look at another one here. And again, this should look familiar. This is another one of the use cases that Srini demonstrated earlier. We have some subject nodes here, which are connected to these projects, which are lung cancer studies. So if we were to look at each of these, we should see in the description that there's some reference to lung cancer in them. And we know that these subjects have been, if I just highlight the neighbors here, clear up some of the noise. We can see that this subject, if I zoom in, was tested for cancer and is associated with Homo sapiens. That's just, we know that this subject is a human. And this one in particular is connected to this. Let's see what the description is here. Some study, novel anti-cancer compound was used to A549 lung cancer cells and so on. So again, we're great that we have these results, but we're interested, how did we get here? So we go back to this page. Again, we just have these 2 lines or these 2 sort of search panels here that we're using. We're not writing any Cypher. Of course, under the hood, it's being translated into Cypher, but we're just using these sort of interactive panels to build the path that we're interested in. In this case, we actually have 2 paths. So we start with these cancer subjects. So we see we have this subject node, and it has been tested for cancer. We know that it's cancer because we're adding this filter here on in this case, it's the DOID. But you could use maybe a name or maybe the description as well to filter if you know what those properties are. And we know that it's a human, so we have this associated with this NCBI taxonomy. And again, we're filtering on the NCBI taxonomy ID. And then we have a second path saying, we have our cancer subject, but give me the projects that contain those subjects. And we know that this is a lung cancer project because, again, we're filtering the description description contains, and then we have lung cancer here. And if we go and search, we should see our results again. So the key point here is that we have this sort of multi-path structure here. So we have two relationships coming into cancer, and then to add a third, we have the second path. Lung cancer contains cancer subject, and you can add as many of these paths as you like. Of course, as the query continues to grow, the complexity grows, and the time it takes to search to gather the matches may take longer. But in theory, you would be able to create a very complex path here, and using this schema that we've developed here. Just checking on time. I'll run through another example here. I'll switch over to this tab. It's another one that I think Srini just briefly went over. We have this, we're finding canine subjects who have been tested for diabetes and have had blood samples sampled from them. So if we sort of look here, this is our subject node. We know that it's a canine because it's associated with this Canis lupus familiaris node. We see that it's tested for diabetes. And then these are just a bunch of samples that were gathered from the subject. We can kind of see, I'll just cut out some of the noise here. We have this sampled. So this sample was sampled from the subject. And then I'll lock these nodes because they're getting a little antsy. And again, if we highlight the neighbors, we can see that this sample was sampled from blood. And it seems like it's pointing at this project over here. It looks like utility of canine diabetes, some sort of study. So again, how did we get here? How do we produce these results? We'll go back to our advanced search. And this time we see that we have 3 paths that we're doing. So start with, this should start to look sort of familiar at this point. You start with a dog subject, tested for diabetes, associated with, call it Canis lupus. And again, we're using all these filters here to find these nodes. Then find me blood samples that were sampled from these subjects that we found. So we find the biosample first, and we filter to make sure that that biosample was sampled from blood. And then finally, go from those biosamples to the study. And we also want to make sure that this study contains both the subject and the blood sample. It's possible that maybe that's not the case, but in this case, we're ensuring that any studies that we find have both the sample and the subject in them. So again, we search, we get these results back here. So let's see, I'll show one more use case quickly, just so I know we're getting close on time, but I'll switch over to this path here. So this one is a little more complex than what we've seen so far. So we're specifically interested in, given a disease, in this case it's leukemia, find the files, any files that reference this disease, and then give me the data type, and then also find me the project of those files, and then we're also getting the DCC, which in this case is links. So again, if we go to our advanced search, we can see that we have this very sort of long set of paths that we're doing here. So again, briefly, we'll start at the top. We want to start with links, give me the root project, and then from the root project are all the sort of sub projects that are contained within the DCC. So we'll go from those sub projects, give me files, and then I want to ensure that those files are some sort of data type. You could filter here. We actually don't have one in this case, but you could filter by data type if you wanted to. In this case, we're just saying, tell me what the data type is of whatever files we found. Then find any subjects that were described by the file, and then filter those subjects on leukemia specifically. And again, if we go back, we can see, give it a few seconds there. We found leukemia, and then some subjects, and then some files that describe them. And then again, just for this file specifically, we can see this project, the links project contains that file. And then in this case, the data type of the project is gene expression profile. So that's what we've been working on. I think cutting a little bit into the demo time, so I'll stop it there. But as Mano mentioned, he shared a link in the chat to a site that we have where this is live. I certainly encourage anyone intending to play around with the search. And I think as Srini mentioned, it is very much in development. So if you run into any issues, please let us know. We're happy to answer any questions. But I think with that, I'll send it back to either you, Mano, or Srini.
Question?
Sure, please go ahead.
Okay, so I curate for the Metabolomics Workbench, and sometimes I'd like to know all the projects associated with a particular organization. Is that something that one can do with this query? Is that part of the metadata?
No,
Yes.
not part of for an organization like the university.
Yeah.
Okay. I believe it is possible. Isn't it, Mano, in the C2M2 schema, you have a metadata for a particular organization?
No, I think in the C2M2 metadata for the project, we include information such as the project title, project description, project ID namespace. But I believe it does not contain any further information like who are the PIs from at which institution that experiment that project was carried out.
Okay. Yeah. Thanks for the clarification.
However, of course, we do have that information available at the Metabolomics Workbench. So if you search for certain keywords, I believe, you will be able to find that. And let me promote, let's see, Dr. Owen Fahid to panelists so that if he would like to provide any information, he can. Owen, I'm going to promote you to a panelist.
Can you guys hear me?
Yeah, we can hear you now.
Yeah, in terms of PIs and all the information about the institutions, we call it institutions, that can be obtained from the Metabolomics Workbench project and study. So now I'm assuming you're going to have the study level and analysis level built into this system too, right? Because most people are more familiar with studies than they are with projects. So there's a hierarchy there, project to study to analysis. So it's a very important component to be able to transfer to this type of schema.
Okay. Yes. So I'm going to talk about development of workflow using a Playbook workflow builder tool, using one of the genes RP as a seed gene to mainly demonstrate the use of tools that we have talked about earlier, such as MedGene and MedNet. So what is Playbook workflow builder tool? Workflow builder tool is a NIH CFDE funded product, and it was spearheaded and led by the links team of CFDE. And Daniel Clark is the lead developer, and it has really gone through tremendous effort. And there are several other data coordinating centers which have contributed to this effort, such as the Metabolomics Workbench, Glygen Data Coordinating Center, Extracellular RNA Communications Consortium, and Gabriela Miller Kids First program. It is a web-based tool to construct workflows using an expanding library of components. It includes meta nodes for entities such as genes, metabolite, and data matrix, and there are processes and transformations that are applied on the meta node, for example, to find more information about a gene or metabolite, or perform principal component analysis on a data matrix. So here is a screenshot of how the workflow builder tool looks like on the web app, where we can see various entity types such as gene input, gene set input, which is essentially more than one gene. And then we have input for other different types such as the protein input and for file input, etc. From the left menu, one can try to filter it out so that you see just a couple of nodes here, and depending upon whether it's related to file or a gene or drug, etc. So I will go into some details and then quickly demonstrate it through an example. So I will be using the gene RP, or ribulose 5-phosphate 3 epimerase, as an example gene. It's a protein coding gene, and as per the gene code information, it participates in the pentose phosphate pathway. The enzyme catalyzes the reversible epimerization of D-ribulose to ribulose 5-phosphate to D-xylulose 5-phosphate. There are some diseases associated with this gene RP, but they are not directly. Instead, based on the literature, it has been inferred that they may be associated with disease called myasthenic syndrome, congenital 10, and retinitis pigmentosa 45. So the use case that I'm going to demonstrate for the workflow builder tool is shown schematically here in this workflow. So we will start with the gene RP and find out what are the other proteins which are interacting with the protein RP by using a string database. Then once we have a list of genes based on the protein-protein interaction network, we can try to do enrichment analysis on that against diseases to find out what are the potential diseases that might be associated with the group of the genes that we are looking in the protein-protein interaction network. One can do enrichment analysis with respect to pathways such as GAP pathway, gene ontology, etc. And we can do enrichment analysis against information contained in provided by the data coordinating center GTEx, which is essentially a collection of the detailed gene expression values for different tissue types. And we can also look at enrichment against CHIA database, which essentially provides a transcription factor target information. And we can also find for those genes what are the reactions, metabolite, and studies in Metabolomics Workbench by using the components from the MedGene tool. And then when we have a comprehensive list of metabolites, we can use some features from the MetNP tool that I demonstrated yesterday to find what are the metabolic pathways associated with that. And continuing, we can apply many other transformations to further expand this network and continue and do further interpretation. So this example has been already posted on the workflow builder tool in the list of use cases provided by the community. So there is one by a similar name, which is a little bit outdated, but nevertheless, this updated one is also available there. And I believe I posted the link to access this use case directly in the chat message. So here, the idea is to identify what are the related proteins and metabolites for the gene RP across based on information from different coordinating centers. So I just want to briefly go over, and I will go into more details as we proceed. The workflow builder tool, as we construct the workflow, it automatically generates a summary of the steps carried out. And I will not be demonstrating in this one, but a more advanced features have been added to workflow builder tool. For example, it can generate a complete report of the analysis, including abstract, methodology, and result section. So the objective is that using this tool, one can produce high quality reports that can potentially be used in journal publications, at least to help in generating part of your manuscript. So we start with the main web page where we see essentially a screen like this. We click on the plus sign to add a node. In this case, I selected gene input from the list and I specify RP as the gene and then click on the submit button. By the way, while I'm demonstrating this, if some of you like, or all of you like, you can go to the website and try it while I'm going through. So I'm going to pause here for a little bit in case you want to go to the actual workflow builder tool using the link that I provided in the chat. So let me wait here just for about a minute so that if you like to try it hands-on right now, you can do that. I can go back one step so that you can try to follow. To start adding a node, you click on the plus sign, and then you will see a list of catalog from which you can select gene input. You can filter the list of those nodes by selecting the checkboxes on the left-hand side so that you will see a short list. You can enter RP as the seed gene and click on submit button. Then once you have entered the RP as the seed gene, the next step is to find what are the other interacting proteins that interact with the protein RP. And for that, we have the transformation called fetch string-db PPI. You click on this, then it will take you to the what appears as here shown here. It will find out what are the proteins and it will list them in a fashion like this. So as we can see that starting the seed node is RP, and then we have other proteins listed here which interact with the RPE. Then we can try to plot this protein-protein interaction network, and for that, so depending upon what the output data type is at each of the steps, if you click on the plus sign, it will give you the, if you click on that output node, it will give you the option to choose from what are the possible transformations that can be applied on that. So in this case, we select the format string-db protein-protein interaction for plotting. Once you click on that, you will see a network as shown here. And in the top part here, you will see that as you proceed, the summary paragraph gets updated accordingly. At the next step, now that we have the string-db PPI, to do further operations, one can extract the gene list from that network. So you select this transformation here, as shown here. Then you will get that in the list of genes have been extracted. And on that one, you can perform further operations. For example, here, we can apply enrichment analysis. That's a tool developed by the Links team. So within enricher tool, it can perform enrichment against many databases such as CHIA 2022, ENCODE and CHIA consensus transcription factors, ArchS4. We don't see that in the screenshot here, but there is, one can do against care pathways and many other pathway databases. Of course, from this page, one can further follow through any of these and find more information. And there is ability to save some of these results text file or possibly other formats. So at the next step, we go back to the gene list that we created, which was essentially the nodes in the protein-protein interaction network, starting with the seed gene RPE. And now we can apply other steps. For example, we can find using the components from the medgene tool, what are the reactions in which those genes might be participating as potentially as metabolic enzymes. So we click on this node here, and we get the list of reactions that are associated with some of the genes. It is possible that a gene or the corresponding protein may not be acting as a metabolic enzyme, so we will not see that. So that's why here we see only 3 genes listed. Continuing further, we can try to find what are the studies which are associated with the metabolites that are associated with those enzymes and reactions. So we can click on that node we saw on the previous slide, and then we will get a list of studies from Metabolomics Workbench, like we see here. And we can click on any of these to find more information or follow through that the page for that study as Dr. Sumana Srinivasan demonstrated yesterday. Then we can find more information or find the metabolites corresponding to those genes. So essentially, these will be the metabolites which are participating in the reactions catalyzed by the enzymes corresponding to those genes. As we can see here, we can follow some of these links, for example, Metastat, to find more details about that metabolites in terms of as it was demonstrated by Dr. Onsai. Excuse me. Excuse me. Then to further for further operations, we extract a list, the metabolite set as a list from the table that we saw earlier. Then we get the list of metabolites. And once we have this list of metabolites, there are some operations that we can carry out. So for example, here we can find more information about those metabolites, such as here we can find, again, all this information is getting extracted from resources at Metabolomics Workbench. For example, we can find the exact mass, formula, and other information for these metabolites. At the next step, based on the list of metabolites, we can perform METNP analysis on the list of metabolites. Now, in this case, we do not have any numerical data for those metabolites, and we did not upload any metabolite data matrix. So this will be essentially performing enrichment analysis and finding what are the metabolite classes for those metabolites, and look at generate a network of metabolic pathways to show what are the metabolic pathways with which those metabolites are associated with this. So this step actually can take about 2-3 minutes to complete, depending upon how many metabolites we have in the list. And if the list is really long, sometimes the program may not complete because we have a timeout limit of 5 minutes on our servers. So I suggest if one want to explore, I start with a shorter list of metabolites. So once this METNP pipeline is completed, the results are presented in a tabular format where we have several types of results. And we saw some of those yesterday, for example, a plot of metabolite counts. Then we have a list of the genes which are associated, essentially corresponding to the enzymes participating in those reactions. Then we have the network corresponding to showing what are the different metabolic pathways to which different metabolites are associated with this. And we have some other information as well that one can explore. One can continue this process and keep on building on these networks, which will help adding interpretation of these processes. So in the workflow builder tool, once you have created a workflow, you have the ability to save and export the results and the workflow in several formats as shown here. For example, if there are any unsaved changes, this button will be, this floppy button will be highlighted. You can click on that and it will give you the option to, it will save it. And then you have the ability to export it in different formats, such as a JSON format, etc. There are a number of other options to export it in other workflow formats. This shows an example of if you click on the export option as a JSON, then one can save it here. And later on, you can actually upload this workflow when you go to Workflow Builder tool, so that you do not have to recreate it. So this is very important for usability of the effort that you have already put. So that essentially completes the demonstration part. I will go to the Workflow Builder tool live and try to just show some of these steps again. And I have provided the links here and some links in the chat as well that you can follow through. So with this, let me go out of the presentation mode and go to the Workflow Builder tool website. So this is the example that is already provided. I can go from the beginning here. You can go to published playbooks. These are the ones which have been curated by the core team, but there are other playbooks contributed by other users. In this one, I believe this is the one. So we click on this one and we click on launch. And you will get this one. One thing I want to highlight is that an interesting feature of this tool is that as the computations are happening, it shows a progress here on each of these steps. And once it has been completed, that entire box will be in a green color. So what I can do is I can to add with the demonstration and give some examples. I can show that example here again. We start with this sign by clicking on the plus sign. Okay, so here I choose the gene input. I specify RPE. And if you enter part of the gene symbol, it will show a list of potential gene symbols from which you can choose. So at this step, I can, of course, there are several of these applicable. For example, I can find the protein-protein interaction network, or I can find additional information by clicking on gene ID conversion tool, which will show the other different types of IDs for that gene, such as anti-gene ID, ref-seq ID, UniProt ID, etc. Or use the MedGene tool to find all the information related to that gene in the context of Metabolomics Workbench related information and metabolic related information. In this case, I'm going to follow the demo that I showed in the slides. So it computed the protein-protein interaction network and it is listing it in this format. Then I can click on the plus sign here and we format it for plotting. So we have the protein-protein interaction network plotted here. For the next step, there are a couple of options. So this is the actual PPI node as a table. So that's what I click on. On this one, I can extract the gene list now. So now we have the gene list. We can continue with the additional operations on that. For example, with the gene list, we can apply here MedGene related tools. For example, with that, about the reactions associated with performing the enzyme. So we're listing quite a few. I think in my screenshot, these on this one. Then we go to the gene set gene list again, and then see what other operations we can do. We can look at other MedGene related components. For example, find the metabolites that are associated with that gene set. We have that metabolite list with a little bit of additional information, such as the compound ID, refinite name, corresponding to reaction ID and metastart link. You can follow through the metastart link to find more information about that metabolite available within the Metabolomics Workbench database. For example, what is the spread of relative standard deviation value across all the studies, and for this, in which study the analysis it appears, and if there are suitable conditions on which it can compute ANOVA, then it will list the p-value and Francisco rate corresponding to that statistical test and other information that Dr. Weinfeld presented on Monday. We can find what are the studies related to the metabolites in Metabolomics Workbench. So one interesting feature of the workflow builder tool is that it learns based on what other users have tried. For example, it noticed that we have already looked at the metabolites and reactions associated with those genes. So it is not listing those near the top. Instead, now it is listing what are the gene studies related to the genes. We can click on any of these to find more information, more detailed information about these studies. Going back to the list again, or rather, we have a list of metabolites. From this, we can extract the list. Once we have this list, we can apply TMP analysis or extract more information. As you saw earlier, we have a lot more detailed information related to these metabolites. From this metabolite set, now we can perform the MetMP part of the MetMP pipeline. So this step actually, it completed very fast because the system has recognized that this step has been carried out on this precise list of metabolites. So that information it is able to pull from the cache. Otherwise, as such, this step could take 2-3 minutes if you start with another gene or another list of metabolites. It's a PDF file. This information is in terms of JSON, which can be used as input to other analysis steps. So that essentially completes the demonstration I had in mind. Of course, one has the ability to let me go to the network report view here. In the report view, we have the option to actually even edit this summary if we want to save it after editing. And here we have the option for saving this, and then we can export it into JSON format or save it as other workflow formats such as by compute object, and export this ability to save it to the shared public library of workflows so that other users will be able to see this use case if you go to the published Playbook. And once you have saved the workflow as a JSON to your local computer, next time if you want to start from that, you can click on this button to import it, and then you will be able to start from that. So with that, I complete the presentation. We'll be happy to take any questions.